Hi everyone, my name is Akif Eskalen. I'm extension plant pathologist in the Department of Plant Pathology at UC Davis. Today, I'm going to talk to you about a new non-native pest and disease complex threat to valley and blue oaks in California. Before I start my presentation, I would like to acknowledge my collaborators from Department of Plant Pathology, UC Davis, CDFA Diagnostic Laboratory, Forest Service, CAL FIRE, Departments of Entomology and Environmental Studies from UC Riverside and UC Santa Cruz, Davy Tree Expert, and Pacific Tree Care Company. Have you seen these kind of symptoms that are dying oaks in your area? We recently identified a new non-native ambrosia beetle, which is Xyloborus monographus from dead or dying valley oaks in Calistoga area. So this is the female beetle, which is very small, 1.8 to 3.6 millimeter length. It is as big as sesame seed. This is male, which is very small, smaller than the female. Most of the ambrosia beetles are associated with fungal species. They develop a special organ in their mouth, which is also known as mycangia, where they can carry spores of the fungal associates. From this particular beetle, we recovered several different fungal species, including Raffaella montetii and Fusarium solani, which I'm going to talk to you about later. The reason that these ambrosia beetles are carrying these fungi is that they cultivate these fungi in their broader galleries, which is in the wood of the plant, and then they feed on these fungi. So they are kind of fungus farming beetles, which is very common some of the ant colonies. Now with the ambrosia beetle, it has a new name known as Mediterranean oak borer. The disease that caused by the associated beetles is known as oak borer wilt. So these beetles are non-native to North America. So when we look at the background, in 2018, the beetle was first trapped in Portland, Oregon. But there is no record of causing any damage on any oak species at that time. In same year, in 2018, the fungus Raffaella montetii was isolated from, again, dead and dying valley oaks from Calistoga by Garbaletto laboratory, but there was no beetle identification recorded at that time. In 2019, both beetle and associated fungi were identified by our laboratory in collaboration with CDFA and also with Dr. Bob Rabaglia, who is a taxonomist from USDA Forest Service from Washington State, and South Hammer Laboratory from UC Riverside. Where is this beetle native to? When we look at the literature, this beetle has been found in Morocco and Algeria in African continent, Turkey, Iran, Korea, and Israel in Asia, and several different European countries, including part of Russia. What are the current hosts in their native area? Again, from the literature so far, what we know is that this beetle has been found on chestnut, beech, and several different oak species, including some of the oak tree species that we have found in California. So I just want to point out that these beetles in Mediterranean region are known to attack or colonize dead or dying or stressed plants. When we look at the current distribution, we first identified this beetle in Calistoga, Napa area. So later on, Forest Service, in collaboration with CAL FIRE and counteract commissioners, they deployed the white sticky traps in different locations to have an idea how widespread these beetles in the Napa and Sonoma area. So these red dots are showing where the traps are located. This one single blue dot is showing where the traps are located in Blue Oak area, which this beetle seems to be attacking Blue Oaks as well. So these black circles are showing that these are the ones that the beetle has been confirmed positively by their morphology and also their DNA analysis. So in this picture, I would like to show you the current distribution of the valley oak in California to have an idea where these beetles potentially could spread in California. So as I mentioned that this beetle is also confirmed on blue oak species. Again, so this is the map is showing the current blue oak distribution in California. Again, these are the potential spread areas for the beetle. We recently also identified the same beetle on single black oak species in Calistoga area as well. So these are the map that shows the very current distribution of the black oak species in California. When we look at the symptoms, it is very interestingly difficult to identify whether these oaks are infested by beetle or not. So because the bark of the valley oak is very rough, you cannot immediately identify what's going on there. So when you look at these symptoms close up, some of the symptoms look like the oozing, some of the staining on the bark, which could be also misidentified with other the fungal diseases of oak species in California. So this is another symptom on one single entry hole, so which you can also see some of the frass is accumulated and then stuck at the entries of the Beetle Galleries. These are the very common symptoms that you can only see on the cracks of the bark. When you peel the bark, beyond the bark, that you can see this kind of discoloration, which is caused by the fungal associations of the beetle. So again, this is another symptom. This is one of the heavily infested the valley oak species in Calistoga area. So these are the dieback. That is very common symptom on oak species. So another common symptom that you can see around the trunk, that accumulation of the frost. Sometimes you can even see the beetles are walking or flying around this area. So these are the very common symptoms on heavily infested oak species. So these are the symptoms in the woody part of the plant, as you can see. So this is the close-up picture of one of the infested oak. So all these discoloration caused by the fungal associations of the beetle, including tree response from the plant. This is another valley oak that has been removed for a while. So I just wanted to show you how the fungal associations of the beetle could colonize on the active xylem of the plant. You can also see some of the beetle galleries in this area. The beetle seems to be attacking younger plants, but younger plants are responding more than the older plants, I will say. In this area, you can see the one attack on relatively younger valley oak. So the plant is able to recover and then block the entry point of the beetle. However, when you peel the bark beyond the infested area, you can still see the discoloration caused by the fungal associate and then entry point right here of the beetle that you can see. This is a close-up picture. You can even see the gallery going through the woody part of the plant. So these beetles seems to be also colonizing upper level of the plant, some of the big branches. So you can see some of the broken branches in heavily infested oak area. So this is one of the example. When you look inside, you can see all the galleries and the discoloration from the fungus. And so some of the broken part of the plant, you can even see the extensive beetle galleries. These are the mechanical damage that makes the branch very vulnerable and breakable, and then within the small wind, they can just break down and then fall down on the ground, which could be a public hazard in heavily infested areas. So this is the beetle galleries that also shows that blackened brownish or whitish area. So multiple fungal species are colonized that were introduced by the beetle. So they are kind of like this. Right now you can see their fungus farming areas where they feed on fungi. This is the individual, the beetle on the galleries. There's another one. These all are the females that you can see. So this next picture is showing how big these beetles comparing with the tip of the plant. That will give you an idea what to look for, what to expect to see. How does it spread? As you may have heard or you may know that the infested firewoods is a source of the transportation of the, not only for this pest, but also other pests. So this particular ambrosia beetle seems to be the continuous staying alive on cut branches for several months until the wood is completely dry out and not be able to support the fungal colonization. So therefore, they could be potentially transported from one location to another location. So that's very important. So they can also fly. They are a Coleoptera group of the beetles. They are not good flyer, but short distances, they can fly from one location to another location. So from the literature, again, so there are a lot of studies has been done in Mediterranean region on these beetles. So this is the one that shows you when the beetle flights are more peak or active. So in this particular literature shows that the beetles could be more active and flying beginning from the March, April, June, July, and the end of the July, that peak is going down. So this is another study, 3 years of the study that shows the similar results. So beetle seems to be active in April, May, June, July, in those years. So what are the fungal associates of the beetle? So when we look at the literature, this beetle fungi has been identified and then described in 1998 by Moralet, which is the Raffaella Montetii in France. Later on in 2004, same fungi has also been identified on Quercus robur in southwest Germany. It's another literature. So another literature in Portuguese, 3 different locations, same Raffaella Montetii has been identified and found from cork oak. In Tunisia, which is interestingly, Fusarium solani, we also found the same species from the valley oak has been found from the pistachio bark beetle in 2019, which is the new the news to us. So in Calistoga, Napa County, from our studies, we have identified several fungal species, including Raffaella Montetii, Fusarium solani, Leptographium. These are the fungi that has been recovered from the Mycangia, wood, and also beetle galleries. We also identified some of the species of the Paracilomycete from the beetle galleries. We are still not sure if these species are specifically associated with the beetle or secondary invaders. We also identified several different yeast species from the wood and galleries of the mycengia of the plant beetle. So this is the fungal colony, Raffaella montegii. So the one on the top that you can see the colony, the one on the bottom is the colonies or the conidia and conidiophore of the fungus, which is very small. So this is the Fusarium solanae. Probably it's a new species which is associated with the ambrosia beetle. So these are the macro and microconidia of the fungus. The question that we still have is that are they pathogen to oaks? As we speak, we are running a pathogenicity test on young, healthy, the valley oak seedlings in greenhouse. So soon we are going to be able to have an idea whether these fungal pathogens are pathogen, or if they are pathogen, how aggressive pathogen they are to oaks. So when we look at the current management, so we haven't done intensive studies on this particular beetle, but we have some information from the other ambrosia and the pest and disease complex that has been done in California, in other parts of the United States. Don't move firewood. It is known that not only for this beetle, but also for other pests, the firewood could be the number one potential transportation method for this beetle. So when you have the firewood, that means these beetles are having transported 70 miles an hour with that speed. Remove heavily infested trees to reduce the population in that infested area. So when the population is so high, so those beetles are going to have better chance, a better successful the colonization in that area. So it's very important to remove the heavily infested trees. I have to stress out that I wouldn't recommend removing any infested trees. Sometimes when the tree conditions are better, they may recover back from these infestations. So that's very important to remove heavily infested trees just to reduce the population. Again, chip infested wood 1 inch size. This is the study that has been done by other invasive shuttle borer group of the pest and disease complex. It showed that it's very successful. When you chip the woods 1 inch size, you can control these beetles 95–96%. I will say if the chipping the infested wood could be utilized in that infested area, you can spread down and then you can utilize in that area. However, if you would like to transport that chip from one location to non-infested area, I would recommend treating them with either solarization or composting them to make sure you are not transporting any beetle from one location to another location. Because what we know is that one single female could colonize and establish a new colony wherever they arrive in these areas. So chemical options are being investigated. We still don't know what chemicals will work. We have some ideas from other studies that we have done on invasive shuttle borers, what to do, what to work. So as we speak, we are now planning to set up a series of the trial to see if we can pinpoint or identify chemical options that could be applied to control these pest and disease complex. So my last point is the report, and any oak borer wilt suspect 3-2, you can report them your county ag commissioner's office. You can also report them the advisors from UCANR in your local area, which you can find their information from ucnr.edu. You can also report them in CAL FIRE. So those are the entities who has been trained to know how to identify them or how to get help you to identify these pest and disease complex. With that, I would like to thank my lab group for our identification of these pests and diseases. I also would like to thank my collaborators from CDFA Diagnostic Laboratory, Forest Service, CAL FIRE, Davy Research Group, and Pacific Tree Care Company. I would like to also thank to funding sources. We recently got the funding from USDA Forest Service Evaluation Monitoring. As we speak, we are trying to establish our grant, so we will be doing more identification and also some control strategies, how to control these pest and disease complex. With that, I would like to thank you for your attention.